Chapter 12, Nervous System, Nervous Tissue, Part 6. All right, so as I mentioned before, hyperpolarization is often caused when those voltage-gated potassium ion channels open, causing the charge to be, say, negative 80 millivolts. And then to reestablish a resting membrane potential, that it requires both the closing of the potassium ion channels and then the sodium potassium ion pumps to function to reestablish both the resting membrane potential and the proper distribution of ions across the membrane so that we have high sodium ion channels on the outside of the cell and high potassium ion channel ion concentration on the inside of the cell. We also have the refractory period, that short period of time in which no action potential can be initiated. First, there's the absolute refractory period. That is when the sodium ion channels are in their closed inactive state. So here they are, closed inactive state. The second door is closed, so nothing can get through. There's no way for that to open. That does not respond to voltage. It just takes a little bit of time for that to respond. And then once the voltage-gated sodium ion channels return to their resting closed state, then we have the relative refractory period where you could potentially get an action potential, but it's going to require greater stimulation because that part of the axon is hyperpolarized. It's hyperpolarized, so it's a greater distance to get to the threshold. All right, here is the synaptic knob. As we know, concentration of calcium ions means there's high concentration outside of the synaptic knob, low concentration inside. There is a calcium ion pump that's constantly pumping calcium ions out of the synaptic knob. But when the action potential arrives, the voltage-gated calcium ion channels open. Calcium ions flood in, overwhelming those pumps. The high concentrations of calcium ions allow them to bind to the synaptic vesicles, causing exocytosis and the release of the neurotransmitter into the synaptic cleft. And then, of course, the neurotransmitters diffuse across the synaptic cleft, bind to the receptors on the chemically gated ion channels, and we potentially have a signal getting passed on to the next neuron or effector cell, the postsynaptic neuron, as it were. Here is a figure in your book, I think it's 1222, that covers everything to try to get you a feel for what's going on in different parts of the neuron. All right, the action potential moves down the length of the axon of the neuron, and its velocity, how fast it goes, is determined by different factors. One is the diameter of the axon. The thicker the axon is, the wider the diameter, the faster the signal is. The other big factor is myelination. If the axon is myelinated, then the velocity is faster. So diameter of axon, larger is faster. Myelination, if you have it, it is faster. So there are two types of conduction, of the traveling of the action potential down the axon. There's continuous conduction. Continuous conduction only occurs in unmyelinated axons. If there is no myelination, then that means you have to cause the opening and closing of voltage-gated ion channels along the entire length of the axon, and that's slower. The other kind is saltatory conduction. This occurs in myelinated axons. It is much faster. Basically, you have the action potential being generated at the neurofibril node. This is where all the opening and closing of voltage-gated ion channels occurs. And then the ions that have migrated across will diffuse down to the next segment. No opening of channels in the myelin sheath. And then the next neurofibril mode will then have its voltage-gated ion channels open because, again, no myelination at the neurofibril nodes. and then again diffusion going to the next section, and so on and so on. So the voltage, the change in charge, the action potentials kind of jumping from neurofibril node to neurofibril node. No movement of ions across the membrane where there's a myelin sheath, just a diffusion of charge down that part of the axon. And so even though the charge from diffusion decreases as it goes down the axon, by the time it reaches the next neurofibril node, it will be at threshold. So as long as it's at threshold, when it gets to the next neurofibril node, you get action potential generated. And this is all, again, faster than the continuous conduction. And then this also is more efficient overall because you need less energy from sodium potassium pumps to maintain the resting membrane potential, because you only need to worry about it at these neurofibril nodes. So this is actually more efficient in terms of energy for the neuron. All right, there are 3 different groups of nerve fibers defined based on whether they're myelinated or not, and whether they're wide or not. The group A is the fastest. Their conduction velocity is like 115 meters per second. How fast is that? Well, I converted it over here to miles per hour. So the charge, the action potential, is traveling at 335 miles per hour. So that is pretty fast. It requires large diameter axons that are myelinated. So we need large diameter myelinated fibers. And this is most common in somatic sensory neurons and somatic motor neurons. It makes sense because they have the longest axons, going from our fingers and toes to the spinal cord, for instance. And then group B and C either have a small diameter, or they're unmyelinated, or both. These are some of the sensory motor neurons for the visceral neurons. It turns out the signals coming from our internal organs travel slower, and this could be 15 meters per second, or for group B, 1 meter per second for group C. Basically because either it's unmyelinated or it has a smaller diameter, or both. And then inside of the brain and spinal cord, they tend to be group B and C too, because they're only traveling tiny, tiny distances. All right, neurotransmitters. There are different kinds of neurotransmitters. One category is acetylcholine. We've met acetylcholine already. We know that it is excitatory when it comes to skeletal muscle tissue, but it turns out acetylcholine can be excitatory or inhibitory, depending on where it is being released. And it is found both in the central nervous system and the peripheral nervous system. So it is a very common, widely used neurotransmitter. Then some amino acids are neurotransmitters. Those amino acids can be excitatory or inhibitory. Then monoamines, these are smaller than amino acids, derived from amino acids. They also can be excitatory or inhibitory, and includes things like norepinephrine, epinephrine, and dopamine. Just so you know, norepinephrine and epinephrine are actually forms of adrenaline. And then neuropeptides. These are short chains of amino acids, can also be neurotransmitters, and that includes somatostatin, for instance. So eventually we have to have the neurotransmitters removed from the synaptic cleft. We need to end the stimulation at some point. Ways to do this include degradation. We can have an enzyme, for instance, come in and break down the neurotransmitter. We know this happens with acetylcholine. So acetylcholinesterase will break down acetylcholine, ending the signal from that neurotransmitter. We also can have reuptake, where the neurotransmitter gets reabsorbed brought back into the presynaptic neuron. So that's transport proteins that'll bring it back into the synaptic knob of the presynaptic neuron. This allows it to recycle that neurotransmitter. For instance, we have transport proteins that bring back in serotonin. And then there are various drugs that can prevent the inhibition, the reuptake of serotonin, causing the signal from serotonin to last longer. And this is one way to treat certain forms of depression. Neural pools. Turns out neurons can form complicated groups. This is especially common in the interneurons in the brain and spinal cord. And these pools of neurons can be localized for specific purposes, or they can be distributed in large regions of the central nervous system, basically with different input and output destinations. So converging circuit, you have lots of neurons, lots of interneurons sending input to a single interneuron, and that single interneuron then sends its signal out to cause the effect. So multiple sensory neurons, for instance, may be sending different information, say from, oh, you see a piece of cake, you smell the piece of cake, you're now tasting the piece of cake, all this sensory information coming in to lead to salivation. Just one random example of convergent circuit. Then we have divergent circuits. In divergent circuits, we have a neuron that got stimulated. It passes the signal on to multiple neurons, which then pass it on to even more neurons that send out the various commands or whatever. So the information is spreading out to more and more neurons. An example of this could be controlling skeletal muscle movements. So say you realize you need to move your leg because you touched something hot with your foot, and then that'll send signals to neurons going, hey, you need to send signals, contraction commands to these large skeletal muscle groups, and then this will send signals out and say, oh no, we need to send these commands out to each of the individual skeletal muscles in that part of the leg. And this basically spreads out the command from a single originating neuron. Reverberating circuits have cyclical repeating effects that then lead to the output. So the input may just be one neuron, but then you get these cyclical repeating signals going around and around till eventually they reach the outgoing neuron. This is thought to be important for continual repeating functions. So for instance, to help keep us breathing when we're asleep, we need to constantly have that cycle of breathing out and breathing in going on, and it helps to prevent synaptic fatigue. Although eventually these cycles can reverberating circuits can get exhausted or inhibited. So it's not a forever thing necessarily. Parallel circuits, where basically you've got the information traveling through different collections of neurons, some going from one neuron to the next neuron to the next neuron to the final output, some going straight from the input to the output, and so on. This allows for the signals to arrive delayed from each other so that there's different timing to the stimulation of the output from similar inputs. And this is thought to be involved in higher order thinking. And that is it for chapter 12.